You've already learned how to do one type of ATPase assay, the malachite green assay. And that assay is an endpoint assay, which means that for each reaction, you allow the reaction to proceed for a fixed amount of time, in our case, usually 20 minutes. And then you add the malachite green mix and then the sodium citrate to measure the amount of phosphate that was generated in that period of time. The addition of the malachite dye stops the reaction. So at each individual reaction, you can only measure the amount of phosphate that was produced in the time that you allowed the reaction to proceed. There are many different types of ATPase assays that measure different things and use different reactions and components to measure the hydrolysis of phosphate. So some of them are endpoint assays like the malachite green assay, and there are also some that are continuous assays. So today we're going to be trying a continuous ATPase assay that couples ATPase activity to NADH oxidation by pairing it with an additional reactant and 2 additional enzymes from the glycolysis and lactic acid fermentation pathways. The enzyme pyruvate kinase converts phosphoenol pyruvate, or PEP, into pyruvate as it converts an ADP molecule back to ATP. In a second reaction, lactate dehydrogenase converts the pyruvate to lactate, while oxidizing an NADH molecule to NAD+. We can follow the loss of NADH over time because it absorbs at 340 nanometers. So we can read our plate at 340 nanometers and watch the loss of NADH over time. We can set up multiple reactions in a 96-well plate and then read them in a plate reader, and that speeds up the workflow considerably. The plate reader can be set to take readings at desired intervals, so for example, every minute or every 30 seconds over the time course of the experiment. So you'll get a readout of ATPase activity over time. This type of continuous assay is particularly desirable in kinetic assays because you can make sure that you're measuring the activity in the early linear portion of the reaction when substrate is not limiting. In an endpoint assay, you have to perform separate preliminary experiments where you vary the time of the reaction to make sure that you are in the initial linear phase for the enzyme concentration that you're using. So even better than just being a continuous assay, the NADH-coupled assay is also a regenerative assay, which means that because the ADP that's produced is rapidly converted back to ATP by pyruvate kinase, the substrate concentration remains high for a long period of time in the experiment. So we will be using this assay to, again, look at the kinetic properties of our purified kinesin proteins. And we can compare the results of this assay to the results we obtained from the malachite green assay. And again, we will vary the concentration of substrate so that we can look at the reaction rates at different substrate concentrations. To demonstrate the NADH-coupled assay, I will prepare and assay one sample of the human kinesin 1 motor domain protein. In our plate, each well will contain a final volume of 50 microliters. A reaction is started when 25 microliters of our enzyme in reaction buffer, along with BSA and whatever other components we are including, such as microtubules, is combined with 25 microliters of reaction mix that contains the PEP, pyruvate kinase, lactate dehydrogenase, and ATP. We'll be performing the assay under a range of substrate concentrations, so we prepare more than enough sample for 8 wells down one column of the plate, and each row of the plate gets a different concentration of ATP. So if I were assaying many different samples, each sample would be pipette 25 microliters of the sample reaction in the same concentration, all the way down 8 wells of one column, and would repeat that for each sample that I'm testing. And then each row of the plate will get a set amount of the reaction mix that contains the different concentration of ATP, ranging from high concentration of ATP in row A down to a low concentration of ATP in row H. Okay, so for this experiment, we're going to be testing the human kinesin 1 motor, which I have here in my ice bucket. I have a sample of the human kinesin 1 that's at 0.334 micrograms per microliter. And I'm going to be testing it at a final concentration of 400 nanomolar in the 96 well plate. and I'm going to add microtubules to a final concentration of 800 nanomolar. And then I'm going to be varying the concentration of ATP. So for 8 wells, I need 25 microliters of my enzyme solution with the microtubules and BSA. I need 25 microliters in 8 wells, so I'm going to make more than enough for that. So I'm going to make 240 microliters, and the concentrations in that tube that I make will be twofold what I want the final concentration to be, because I'm going to be adding 25 microliters of that with 25 microliters of the reaction mix that contains the PEP, pyruvate kinase, lactate dehydrogenase, and the different concentrations of ATP. So then I'll be able to combine the 25 microliters of my kinesin with microtubules to the 25 microliters of the ATP concentrations to start the reaction. So to calculate how much I need, I have my human kinesin 1 motor, which, as I said, my stock tube is at 0.334 micrograms per microliter. The molecular weight is 56 kilodaltons. So I can calculate how much I need for 240 microliters. That's twice the concentration of 400 nanomolar to start, with the molecular weight of 96 kilodaltons and the concentration of 0.334 micrograms per microliter. And I see I need 32.2 microliters of that in the 240 microliters that I'm going to make. For the microtubules, I have a stock tube of 1.2 micrograms per microliter, and the molecular weight of the microtubules is 110 kilodaltons. So if I want 800 nanomolar final volume, and I want twofold of that in my mix that I'm making now, 240 microliters of that, the molecular weight and the stock concentration, I see that I need 35.2 microliters of that. I have BSA stock that's 2.5 micrograms per microliter. I want that to be a final of 0.125. So I want in my 240 microliter tube, I want it to be 0.25 microgram per microliter, so twice the final concentration. So that's a tenfold dilution, so I'll put 24 microliters of that, and then I have a 4x reaction buffer. So how I'm going to set up my 240 microliters is I'll have the 32.2 microliters of the kinesin, 35.2 microliters of microtubules, 24 microliters of BSA, 60 microliters of the 4x buffer, and water up to bring it to 240 microliters. Once I prepare that, I'll be able to pipette 25 microliters into each of one column of wells of my 96 well plate, and then I will add my ATP mixes that I've already prepared here that range from 3,000 micromolar to 10 micromolar, which is twofold the concentration that I want final. These are those ATP concentrations in a mix that contains the PEP, pyruvate kinase, and lactate dehydrogenase. So these are ready to add as soon as I prepare and add my reaction. So I'm just going to make my one tube here. I'll start with the water, 88.6 microliters of water. $60 microliters of 4x reaction buffer. $24 microliters of the 2.5 mg per mil BSA. $35.2 microliters of my 1.2 mg per mil microtubules. and 32.2 microliters of my kinesin 1 protein. That's at 0.334 micrograms per microliter. Okay, pipette up and down well to mix. Give it a little flick here. There's some bubbles, so I'm just going to tap it to try and get some of the bubbles out. And then one very critical step of setting up your plate is we're going to put 25 microliters of this into each well of one column of the plate. And it's really critical that you pipette accurately so that they all have the exact same amount of volume. If some have 24 and some have 26, it's going to skew your results a lot when you're trying to plot your reaction rate versus your ATP concentration if your volumes are messed up. So this is one time in particular. In many experiments, pipetting accurately makes a big difference in the results that you're going to get. But particularly in this experiment, one whole column of data really depends on each other, that the accuracy of that pipetting of that column is done consistently. So I'm going to, again, mix one more time and pipette 25 microliters. Into each well of the number 12 column. Okay. And then I'm going to pipette 25 microliters of each of these series of ATP in with the reaction mix into the corresponding rows A through H. Open up my tubes. So for this, I'm going to use a special pipetter that is a multi-channel pipetter that has an adjustable tip format here. So I can go into tubes that are in an Eppendorf rack and then adjust the distance between the tips to pipette into the 96-well plate. This step would normally be done for you by GSIs or staff once the entire plate is set up with each group's reaction. So that the ATP is added all at the same time and then gotten into the plate reader promptly. So I'm going to add the ATPase mixes, and then as soon as I add it, I'll pipette up and down several times to mix, and then I'll take you over to the plate reader and show you how putting it in the plate reader. Okay, so I'm here at the plate reader and I've got the program set up. It's going to just be reading column 12 from our 96-well plate. It's going to be reading for 20 minutes total. It's going to take readings every 30 seconds at 340 nanometers. So I open up the door. I make sure that there's no bubbles in my wells. Put the plate in. Close the door. And then I can press start and we'll watch what happens. So in this view, it's going to show us the readings. That's the first reading at 0 time of each of the wells. And if I highlight up to 10, so I can highlight all 8 of mine, it'll chart the absorbances over time. So we can watch that and see what happens. Here comes the 30-second time point. Okay, so now the run is done, and you can generally see some the slopes here ranging from A12, which is this line here with the steepest slope, down to H12, which had the lowest concentration of ATP, which is this yellow line here, which has the most shallow negative slope. And all the slopes are negative because the reaction is causing a loss of NADH over time. And so what we can do is plot these with Excel and look and calculate the reaction rates by looking at the loss of NADH over time. So it'll automatically open up an Excel sheet that looks like this, that contains information about the run and the data, and it's formatted so that it's showing all of the wells A1 through H12. So our data is only in each of the 12 columns, so A12, B12, C12, D12, all the way to H12. And each row represents one time point for the time 0, and then every 30 seconds to the end of the run. So if we now switch to looking at an Excel file from a full 96 well plate, there will be 96 columns of data. So as you can see, it goes all the way out here to well H12, which is in column CU. So there's a lot of data here, and each plate run will produce a file that looks like this. So for your assignment, when we ask you to look at the results, we will have compiled all those data files from the multiple plate runs into one file for you to analyze, and it'll look something like this. There'll be multiple tabs down at the bottom for different sheets. The first sheet will be a plate setup guide, and it will be a depiction of how the 96 well plates in the experiment were laid out. So in this particular example, 2 plates will be run, so that overall we can have 4 replicates for the SC Syn 8, representing 4 potential work groups, 4 replicates of the CT synate, 4 control reactions with no protein, and 2 replicates of our positive control, which is the human kinesin 1 motor domain. And for each of these samples with enzyme, with protein, there's 2 columns that are run, one for minus microtubules and one for plus microtubules. The reason we run a whole column of samples for each reaction sample is that we will be varying the concentration of substrate. So in the plate, all of these wells in one column will get the same amount of protein, but they'll get different amounts of ATP, with the highest concentration of ATP in the rows A down to the lowest concentrations of ATP in rows H. So in this example, since 2 plates were run, there'll be 2 data files that are the results of each of the 2 plate reader runs. So in this case, we have the data for plate A and the data for plate B. And these look essentially like the files that I showed you earlier. However, we've gotten rid of the information at the top that talks about when the sample was run and the wells and all that information that comes specifically from the reader. And we've added some data labels to help orient you that correspond to the plate setup guide. So for example, in plate A here, we have the 4 SC groups, each with a minus and a plus column, and then 4 no proteins. So in the plate A file, again, you'll see the minus and plus for the 4 SC groups, and then 4 no protein samples. And that's for A1 through 12, so the A row, and then it repeats again for the B row, the C row, etc., down all the way to the H row. At the bottom, underneath all of these data points, there's a row that's for slope, and this is calculating the slope of this loss of NADH over time, and it's over a selected time period. And that will usually be the same for all the columns of data here. And one thing to keep in mind when you have your data here, you might want to plot some of the slopes. So for example, this is plotting all of the row 1. This is plotting all of the row 2, etc. So each of the columns all the way down, it's plotting each of the different ATP concentrations. And you can get an idea of what the slopes look like over time. So you should see a loss of NADH over time, so a negative slope line that will change the amplitude of the slope depending on the concentration of ATP. And you'll notice that in some of the examples, there's a little bit of kind of a period of not very stable loss of NADH concentration. So sometimes you'll see there'll be a little blip up here before things settle down and have a more even slope in the middle. Sometimes you'll even see something weird like this, where it goes up for quite a while before it starts settling down. Or sometimes you'll see little irregularities somewhere in the middle. So you kind of just want to look at that so you have an idea of this part of the time course that you want to take that has a nice, consistent negative slope. So for these examples, I took the slopes between 300 seconds and 1000 seconds. So basically right in this range here, where in almost all cases, anything that was happening kind of strangely here at the beginning gets taken out, and we're just looking at the slopes here. And that's what these slopes in this equation represent. So it's from 300 seconds to approximately 1000 seconds. But if you had a particular slope that had something weird happening in the middle, you might need to adjust the slope here to account for that. So then below those slopes, there's another grid here that again is color-coded and labeled for you so that to help orient you. And this is just taking all these slopes that are here and putting them back in the same orientation of the 96 well plate that they were in the plate setup guide. So this whole row A, columns 1 through 12 are the same here, because these are the first 12 samples, but then here, B1 goes here. So it's just putting them back instead of this long list here that you have to scroll through. It's just putting them back in a 96-well format so you can look at them all in one place and get an idea of how your slopes are changing over the different concentrations of ATP. So that's the same for however many plates of data were run in our particular experiment. Then there'll also be some kind of tab here that will allow you to further analyze the results. So for example, in this case, there's a final tab called Kinetics Analysis. And here, all the samples from the 2 plates that we just showed you here are compiled all here along the top. So it's showing the concentration of ATP that were from rows A through H. And then it's just pulling the data that were from these boxes here. It's pulling them over and putting them all together in one spot. So we have, this is basically plate A here, and then we have plate B here. So we all have them on one sheet. So then below that, we'll have a place for averages of the replicates to be calculated. This may or may not be done for you depending on what the assignment asks, but this will basically be a place to take the, for example, in this case, all of the 4 replicates of the SC synate minus microtubules and average them. And then here's all the SC synate plus microtubules and averaging those. And then over on the right, you would be calculating the standard deviation of the 4 replicates for a particular sample. Below that, then you want to subtract the background of the no protein controls. So that's what this is doing here. It's just for each sample above, it's taking the average and subtracting the average of the no protein background. And the same for the standard deviation. It's subtracting the background standard deviation from each sample's standard deviation. So then below that, there will be places for you to do further analysis. So with this information here and following the guidelines that are in your lab manual, you can calculate kinetic properties such as the ATP turnover rate, the specific activity, and the reaction rate in micromoles per minute. And you'll be using the specific concentrations that were used depending on the semester, what it says in your lab manual or what we tell you in class that you'll be able to use to calculate those. And then you'll be able to use these reaction rates and at the different concentrations of ATP to calculate Km and Vmax values, and then use these values to determine Kcat.